So let's fill that in on the notes. When the nerve impulse reaches the axon's end, the following occurs. Well, before we can understand what occurs, we have to go back to chapter 3. Remember that when you have a membrane to be polarized or resting, ready to do work, it's going to be slightly negative on the inside and slightly positive on the outside. And so this membrane, this muscle membrane, is slightly negative on the inside, slightly positive on the outside. When the calcium comes into the axon terminal, acetylcholine is going to be released. Acetylcholine will come across the cleft and will open up sodium channels. Remember, sodium ions are positively charged, and so they are going to go across the cleft. They're going to go into the muscle cell and cause the cell now to be positive on the inside, negative on the outside, which is called depolarization. So remember, a polarized membrane is slightly negative on the inside, slightly positive on the outside. It has the potential to do work. When calcium channels open and calcium moves into the axon, this causes the acetylcholine vesicles to fuse with the membrane and release acetylcholine into the cleft. And so here you can see them fusing with the membrane, releasing into the cleft, and what they're going to do is they're going to go and attach to the receptor molecules of the motor end plate. So acetylcholine diffuses across the cleft and binds to the acetylcholine receptors on the motor end plate. When it attaches on the motor end plate, it's going to cause sodium channels to open. This is a chemical gated channel. So the acetylcholine is the chemical key that opens this channel. And so what happens is that sodium rushes in. So now you've got more positives on the inside and less positives on the outside, so it's negative on the outside. So this membrane is depolarized. If this is an impulse, and if it's strong enough, if it reaches threshold, it will move down the whole sarcolemma, opening up more sodium channels and propagating or conducting the action potential. So when the electrical signal is, and when it propagates, electrical signals open up the channels, that's called a voltage channel. When a chemical opens it, it's a chemical channel. So acetylcholine opens up this channel, it's called a chemical gated channel. But later on down the membrane, see, this is a chemical gated, and then this is because there's an electrical change. All these will start opening because of an electrical change. The impulse or the action potential propagates down the sarcolemma, down the T-tubules, causing calcium channels to open in the muscle now. When calcium channels open in the muscle, we know what's going to happen. The second stage is the excitation coupling or contraction coupling. And so the calcium is going to go hook onto the troponin, push the tropomyosin down. And if the myosin head had ATP and is in a cocked position, it will connect to the actin, will pull the actin towards the M line, and you'll have a contraction. The action potential is very brief, only a couple milliseconds, while the contraction of the muscle lasts hundreds of milliseconds. The reason why is that nerve impulse or that action potential caused calcium channels to open in the sarcoplasmic reticulum, and it's going to take a few milliseconds to get all of that calcium back into the sarcoplasmic reticulum. So the muscle fiber contracts for longer than the excitation from the neuron because calcium has to be pumped back into the sarcoplasmic reticulum. The action potential also causes slow potassium channels to open, and so now potassium, which is also positively charged, will rush out of the cell, and you'll establish negative on the inside, positive on the outside once again. So how do we stop that message from the nerve to the muscle? Well, there's a couple of ways. One way is that the acetylcholine can just diffuse away from the cleft. Another way is that we have an enzyme called acetylcholinesterase that breaks down acetylcholine, and actually part of it, the choline part, will be absorbed and used to make more acetylcholine in the nerve cell. So this is a picture in your book that shows those steps. And so be prepared on the test to be able to put these steps that happen at the neuromuscular junction in order. Here's another way. This is the neuromuscular junction, what happens, and this is what happens in the muscle during the excitation contraction coupling. So we looked at calcium, didn't we? Remember, in the bone, calcium made the bone hard, and now we see in the muscle, calcium uncovers the binding site. We see in the nerve that it helps us to release the neurotransmitter. So calcium is pretty important. But remember, calcium in bone was hooked to phosphate in a chemical called a hydroxyapatite, and that's what made the bone hard. Well, there are a lot of phosphates in muscle too, and we don't want the calcium to hook up with the phosphate because we don't want hard muscles. They would not be able to slide and contract. And so we have some carrier molecules or some binding molecules that bind up the calcium so it won't mix with the phosphate during relaxation. So the note says the role of calcium ion in muscle contractions. Calcium must be kept in low amounts in the muscle except during excitation because it would bind with phosphates to form hydroxyapatite crystals and the muscle cells would die. So the two chemicals that hold on to calcium and regulate its release are calsequestrin and calmodulin. If you sequester a jury, you keep the jury. So calsequestrin is keeping the calcium, and then calmodulin is moderating how much can be let loose. So maybe you'll remember those two names that way. So we've kind of looked at a microscopic level. Now we're going to kind of back off and say how the whole muscle, what we see in a whole muscle contraction on the organ level. The principles governing a contraction of a single muscle fiber and of the whole skeletal muscle consisting of a large number of fibers are pretty much the same. So if we were to take one little fiber out and hook it to a machine called a myograph, we could actually see what happens in the whole muscle by studying that little muscle fiber, and we're going to do that in a few minutes. When you're looking at the whole muscle, sometimes when you send a muscle contraction, there is sliding, there is contraction, a tension is forming in the muscle, but you may not actually have a movement. So we're going to look at when you have a movement and when you don't have a movement in the next slide. Let's fill in this slide. The force exerted on an object by a contracting muscle is called muscle tension, while the opposite force exerted on the muscle by the weight is called the load. You recall in Chapter 10 we talked about lever systems, and so the effort or the force was caused by the muscle, the load was the bone, and the fulcrum was the joint. So this is basically the same information. When a contracting muscle does not always move the load, it shortens. The tension develops, but nothing is moved. Like if I push on a wall, my muscle is contracting, it's tight, but I can't move the load. That's called an isometric. The load doesn't go anywhere, it stays the same distance. But if the tension develops and the muscle shortens and contraction occurs and there is movement, that's called an isotonic contraction. And muscle fibers in a single motor unit are not clustered together but spread out through the muscle. So that means that this one and this one will be contracting, and then these three will be contracting together. And this is so you don't have like just the top part of your muscle contracting, but it appears the whole muscle is contracting because they're spread out sporadically. So stimulation of a single muscle fiber causes a weak contraction of the entire muscle. So let's look at how we can study the muscle contraction of a whole muscle by looking at a fiber. To investigate muscle contraction, scientists attach muscles to a myograph and record the mechanical contracting activity. The response of a motor unit to a single action potential of its motor neuron is called a twitch. So if I excite just one neuron and I'm seeing how it affects one muscle fiber, that's called a twitch. A twitch actually has three phases. This phase is the dark blue. This is when the stimulus is being sent, but there's no contraction. It's called the latent period. Your calcium really hasn't started to be released. When the calcium is released and we uncover the binding sites and the muscle starts to contract, that is the contraction period. And then when the potassium channels open to cause repolarization and the movement of calcium back into the sarcoplasmic reticulum, the muscle relaxes, so that's the relaxation period. Calcium returns to the sarcoplasmic reticulum, tension decreases, and the muscle returns to the original length. A twitch may be strong or weak depending on the motor units involved. So if I want to say, well, what's the difference in me using muscles to lightly touch you or just to knock you out? It can vary because of two reasons. One reason is that we are activating more and more muscle fibers in motor units. See, there's no activation here, so there's no contraction. Here we only have 2, it's a weak contraction. Here we have 5, it's a stronger, more, until you get the maximal strength of that muscle. So a twitch may be strong or weak depending on the number of motor units activated. Another way to increase the strength is frequency, to hit the muscle without a lot of delay between it. And what happens is that the muscle doesn't completely relax before it hops up and contracts again. So instead of going all the way back down here and going to the same level, it's just going to here and then back up higher. And that is called a graded response, because we're adding these together to get that ultimate response. So the way to increase the strength of a muscle contraction is to activate more motor units, or to increase the frequency on one motor unit. If I just clap my hands in a room, it doesn't make much noise, but if everybody claps in the room, it makes a lot of noise, and so that would be activating more motor units. Now, if I just clap my hand like every 30 seconds and compare that to with very little space in between it, that response is going to be greater, because I'm hitting it with more frequency. Think about when you have a slight headache or a throbbing headache. A slight headache would be maybe where you're sending the impulse every 30 seconds. A throbbing headache would be where you're sending it every second. So 2 ways to get a higher response or